Yeah, so our group is interested in studying the role of the immunoglobulin receptor in malignant B cells, and the receptor is actually is a major target of therapies for the treatment of chronic lymphocytic leukemia. So our group is using preclinical models, mainly animal models, where we try to modulate the expression of the immunoglobulin receptors in models of aggressive and indolent forms of B-cell tumors, and try to understand really what happens when we interfere with the function of this critical receptor for the life of B cells. So this becomes very relevant now in an era where treatments of chronic lymphocytic leukemia and other B-cell tumors is actually concentrating on this specific signaling axis that starts from the B-cell receptor and goes down to kinases that activate finally proliferation and survival of the cells. What we basically recently showed in this preclinical model is that if we interfere with the function of the B-cell receptor in tumors, in tumor cells, we surprisingly saw that some tumor cells resist to the inhibition, so these cells continue to grow, and this becomes a warning for therapies that are actually based on drugs that are inhibiting the signaling pathways. So having identified these cells that can survive the lack of the immunoglobulin receptor, we went on in understanding how these tumors can adapt to the loss of this B-cell receptor and try to identify potential vulnerabilities of these cells, so that if in patients we will have cases where tumor cells will lose the B-cell receptor, we may already help them to identify drugs that can interfere with the growth of these resistant cells. The work that we have done so far in the lab has mainly concentrated on preclinical models. Now, we have identified a number of mechanisms through which malignant B cells can overcome the dependence on the B-cell receptor signaling pathway. Now, the first step is to validate whether those mechanisms that we identified in preclinical models are also present in real tumors, in tumors taken from patients. And we have already identified a number of mechanisms that we identified originally in our tumor model have been confirmed in tumors. So I think the first results are actually telling us that our preclinical studies are relevant for human cancers, B-cell tumors. And now we are interested in understanding whether some of the pathways that we identified are important for cells that are resistant to BCI inhibitors, whether those pathways are also relevant to ensure the growth of human B-cell tumors that are resisting to ibrutinib and other drugs that are targeting the B-cell receptor. One important message that I try to convey to the audience is that we should be careful in giving to patients drugs that are interfering with the B-cell receptor signaling pathway before upfront, before the beginning of the treatment, whether some cells in the patients have already selected mechanisms that will avoid actually the sensitivity to the drugs. So we are trying to identify a number of biomarkers that allow us now to predict whether a patient will or not respond to BCI inhibitors. And I hope that this was a message that passed through my presentation, because it becomes relevant in order to avoid unnecessary therapies to patients that will not respond to the drugs.